talk, and to thank Churchill College for providing many things, among them housing, which is where I am now, and then also this opportunity to deliver this talk. It's on a topic I'm very interested in. Just a super quick background on me. I'm a researcher, and I've been working at the bench and in the lab, working with CRISPR, which is a genome editing tool I'll talk about in just a minute, for several years now. But sort of in addition to that, I've been thinking about the ways that CRISPR can be used, and the ethics around that, and how countries have responded, scientists have responded, the community has responded, and also sort of the lack of interface between the public and scientists. So this talk is aimed at a general audience. If you don't know anything about biology, that's okay. Hopefully what I'm talking about will be as base level as possible. If you do know about biology, some of it might be a little familiar to you, but the ethical parts, I think, are the parts that are most interesting. So without further ado, let's get into it. So the first thing I just want to mention is that a couple of years ago, my colleague and co-author Ikumi Ajedamobi and I wrote a chapter for Springer on this subject. So a lot of what you'll see here comes from that textbook chapter, and so if you're interested at all, please do check that chapter out for full references and a lot more depth on a lot of the topics that I'll get into in just a minute. So to just cover the basics of the biology, because I think this is really important in order to understand what it is we're talking about when we talk about CRISPR. So proteins, which I think are really the key building block of how cells work, how our bodies work. We want to think about these as these tiny machines that do all the important functions in your cells. So for example, these are proteins are machines that bring things from one place in the cell to another. They provide structure to the cell, and they make things, they grind things up and sort of function as garbage disposals. They're really the key building block of the cell. And in order to make a protein, we need something called DNA, which you've probably heard of before. Now, DNA is sort of like the blueprint that serves as the instructional guide for how you make a protein. So with that sort of understanding, and DNA is made up of a series of letters, ATGC, which you may have heard of in other contexts. And these letters and the order that those letters are in determines what the protein does, how it's built, how it's structured. And what's important to note is that if we make any changes to DNA, those changes are permanent. So if we in any way get rid of letters, add letters, etc., that affects what protein gets made in the end. It can make that protein broken. It can increase that protein's activity. It can change what that protein does. There's a lot of things that you can do if the DNA is changed. And so it's important to just understand that basic concept. And so with that in mind, with the idea that if you change the DNA, you change the protein. That brings us right into CRISPR. And it's important also to know that each section of DNA that codes for a protein is what we call a gene. So if you've ever heard anybody talk about genes before, what they're talking about is the DNA that makes a protein. And so what's CRISPR? What's CRISPR? How does that fit into this picture? So CRISPR is, we can think of this as sort of a text editor for DNA. By using CRISPR, we can change those letters that I mentioned before that code for proteins. And it was discovered in 2012, so it's a fairly new sort of discovery. The basic way it works, the details of this don't really matter for the purpose of this talk, but I think it's important to know sort of what we're talking about when we say CRISPR. So the way CRISPR works is that another protein called Cas9 can bind to DNA, cut it, and so that's what you see here. And when it cuts, you can kind of think of it as like scissors. So what it's doing is cutting the DNA into two pieces. And when that cut happens, you get this little gap. And once that gap or that break is made, what we're able to do as scientists is add letters to the DNA, subtract letters from the DNA, or even substitute letters in the DNA. And each of these processes, each of these changes we make to the DNA will again change the protein in some way or another. And this provides an incredible amount of potential. This was revolutionary when it came out. It's still revolutionary today, because it allows us to study genes, how they work. If we make change A versus change B, how does that sort of affect what happens to the protein? If we apply the specific genetic code of one person who has a disease versus somebody who doesn't to a protein, what is it that changes about that protein that's made from that DNA that's been changed, that is why this person has a disease in the first place. So it opens up an enormous amount of potential. And in fact, CRISPR has already been used in a variety of organisms, way too many to list here, but broad categories. Plants have been edited both in the lab and out. Animals for livestock and also for research purposes. Bacteria, and then of course, also in human cells. So in the lab, like the lab I work in, which is a cancer research lab, we use CRISPR all the time to take a gene away and see what does that do to the cell? Does it lead to cancer? Does it lead to other changes that we can then monitor? So what's the problem with this, right? This sounds great. It is great. It's very useful. It's changed genetics research and research across many, many, many fields in many ways for the better. It's really just facilitated all of our experiments and allowed us to really look at it in a very specific way at what proteins are doing and how changes in those proteins might lead to things like disease or structural changes in the cell or whatever the case may be. So as we start to think about the problems with genome editing, I'm going to just give you a couple of buckets, couple of different ways to think about how different ways we can edit the genome and what those consequences would be. So the first is therapeutic. So as I mentioned before, we might use this in a research setting, but we also might use this in an actual person, because you can imagine that if we edit the genome of somebody who has some sort of illness, particularly a fatal illness, if we can change their DNA in such a way that we can save their life, that would be a therapeutic intervention. And this is what I think a lot of people think of when they think of genome editing, and it's very, very exciting. And this is probably the least controversial of the options, although it even in itself has some controversial consequences there. The next is preventative. So this is if we make a change to DNA that isn't saving a life necessarily, but is sort of preventing the chance of that person getting an illness down the road. So if we give them some sort of edit that prevents them from getting a heart attack down the line, for example, that's preventative. So it's not saving their life because they don't have any heart disease right now, but they won't get one, or they're less likely to get heart disease down the line because we've made a change. So that's preventative or sort of prophylactic genome editing. And then there's cosmetic. So these are things that are purely just so you can, for example, increasing your muscle mass or something like that. That is sort of the last bucket. And these last two buckets sort of fall into a broader category of enhancements, so-called enhancement. You'll see this in the literature a lot. And as to whether or not that refers to preventative or cosmetic editing, it's a sort of a vague concept, but this should give you a good idea of the kinds of things we can do with CRISPR. So just one example of each of these kinds of genome editing. The first is therapeutic editing, genome editing. In this case, we're looking at a disease called Tay-Sachs, which is quite an unfortunate illness that manifests a few months into a child's life, and then by age, it leads to motor function loss and a lot of other complications, and then leads to death by age 4. So it's a very, there's no cure for it that we have at the moment. And so there's some literature out there about how we might use CRISPR, for example, to help correct this change in children with Tay-Sachs early on in life, perhaps in the embryonic stage. An example of preventative genome editing would be PCSK9. So earlier when I said that we could edit a gene in order to prevent you from getting heart disease later in life, that's not actually an abstract concept. This is a real thing. So PCSK9 is a gene that if you turn it off, you decrease your risk of heart disease by a dramatic margin. So this is something that a lot of people are looking into, and it's already actually in development for drugs to affect PCSK9 in a non-permanent way, in a non-CRISPR related way. But there's a lot of interest in doing it in a CRISPR related way so that you have permanently sort of protected from heart disease, or at least your risk goes down dramatically. And so that's what this is an example of preventative genome editing. But again, this isn't for somebody who is suffering a heart attack at the moment or has heart disease now. This is sort of a preventative action done earlier in life. And the last is cosmetic genome editing. So this cow might look to you like a Photoshop. It's not a Photoshop at all. It's in fact a real cow. It's called a Belgian Blue. It's a breed of cattle found in Belgium. And this cow's not been edited with anything, but it features a mutation in a gene, again, a gene that codes over a protein. The protein is myostatin. And that protein controls whether or not you stop making muscle, basically. That's the concept there. And so because these cattle have a mutation in myostatin, they don't stop making muscle. They just keep bulking up. And you get this sort of almost comical look in these cattle. But you can see how this might be used in not only other animals, which has already been done and demonstrated that this can be done in rabbits and sheep and pigs and others, but in humans for things like sports or for cosmetic reasons, you can see how somebody might think, oh, well, we could just turn myostatin off and bulk up like crazy, right? So that would be a purely cosmetic change, right? There's not really a good medical case for why you would want to do that. So this brings up a whole variety of issues, as you're probably thinking of yourself. And just one of these, so there's a lot that we could get into, but we're limited on time. So I'm just going to focus on a couple of basic ideas. So one is that not everyone wants to be fixed. It's from an article in The Atlantic in 2013, which is that it sort of touches on the concept that Silvia Camporesi, who's a bioethicist who's talked about a lot of these ideas, and you'll see her name pop up throughout the presentation, is that because there are these communities like, for example, folks in the deaf community, folks in Down syndrome community, who have built sort of their life, their identity, their culture, and other aspects of just living around these differences. There's a lot of pushback from these folks around the idea of genome editing. So for some of us, we might think, oh, well, deafness, that's something that needs to be fixed. But for folks who are deaf, that's not something that's widely accepted. And so actually what you can see here is this is a screenshot of a show called Undone. And in that show, the main character is deaf and gets a cochlear implant, which isn't genome editing, but it would function in a similar way. In the sense that it sort of fixes deafness. And there's a lot of controversy around cochlear implants already. So you can imagine that if somebody were to say, I'm going to edit a deaf gene out of the population or out of this individual, that there would be a lot of pushback there. So this is something really to keep in mind and to sort of bear in mind as we start talking as scientists interface with the public on issues of disability and issues of genome editing in that context. So now, it's important we're going to go back to biology for just a minute. My apologies for those who find biology inscrutable, but we're going to just tap into this very briefly, because it's important so you can understand how regulations are made around these technologies. So we're gonna dive into these big words here: somatic, germline, and see what they mean. So what are somatic cells? So somatic cells are almost all the cells in your body: skin cells, liver cells, brain cells, kidney cells, bladder cells. These are all somatic cells. These are not what we call sex cells like sperm and eggs. Anything that's not sperm and egg, that's a somatic cell. And why is it important to understand what somatic cells are? Well, if we take a skin cell and we edit it with genome editing, with CRISPR, then the product will be an edited cell, but that cell will not go to your offspring. It will not be inherited by your offspring. If I edit your skin cells, your children will never if you then have a baby, that baby will not have any of those edits that you've made. However, germline cells, which are egg and sperm cells, unsurprisingly, because these are what make babies, if we edit germline cells, your offspring will inherit those edits. And this is where things get a bit dicey, right? Because you haven't just made a decision for yourself. You haven't just decided you wanted to bulk up with a myostatin edit or prevent heart disease or whatever. You've made that decision for a future generation. And so this is where regulatory bodies have sort of drawn a line in the sand, and they said that because we have to protect the consent of future generations, that's sort of the phrasing they use in these documents and these regulations going back decades, well before CRISPR existed, but sort of with the concept that we might one day be able to edit the germline. Because we want to protect the consent of future generations, germline editing is now forbidden. It is not allowed, and it's something that we oppose. A lot of governments have sort of made this statement, and a lot of regulatory agencies have operated under this germline versus somatic difference. And actually, this is a paper that looked at regulatory policy and legislation around the world. And this is from 2016, so some of this may have changed, but you get the general idea here that on the right-hand side, you see somatic editing is intermediate. So that's sort of not quite restrictive, but it's not permissive on somatic gene therapies, but that a lot of those same countries are much more restrictive on germline gene therapies. Now, again, some of this may have changed in the intervening years, but you get the general premise that that's how these countries sort of see the differences between germline and somatic cells, that they're considered to be quite different. But is it really that simple? So this is something that Silvia Camporesi has spoken about, and I think it's a great point, which is that we actually have the ability, the technology somebody got a couple of people got the Nobel Prize for this, to turn a somatic cell into what is known as a stem cell. And that stem cell can then become either a somatic cell or a germline cell, into a sex cell, a sperm or an egg. And if we can do that, then can we also turn a somatic cell into a stem cell and then turn that stem cell into a whole organism? And in fact, that's been done. That was done in 2009, well before CRISPR existed, with mice. You can actually see these mice here. These are mice that were created entirely with stem cells made from somatic cells originally. So where was the line between what was somatic and what was germline? And if you had edited those somatic cells before you turned them into stem cells, have you not edited the germline in a way? So this is kind of an interesting wrinkle in that problem. And it raises questions, of course. The first question is, well, can you do this in humans? Well, it hasn't been done. So just to not to worry anyone, this process has not been done before. There's a lot of regulations that sort of protect against this, but it does sort of raise this question, and particularly in the context of, for example, IVF, which is highly regulated. But if you were able to turn somatic cells into eggs or sperm, then you could potentially use those through in vitro fertilization to produce a baby. And if there's any editing that goes on at any stage of that process, you've now edited the germline, even though you maybe technically didn't, because you started with skin cells and you only edited the skin cells that then became sperm down the line. So this is the kind of thing that we're sort of thinking about when we talk about the difference between germline and somatic, and how that's sort of a blurry line. Then there's this other issue of epigenetics, which I don't want to get into too much detail at all on, but just to say that there's this, so we know that the letters that make up DNA control or dictate sort of what the proteins are that are made. But there's also how those letters are read, and there's a whole process, which I won't get into, of how that happens in the cell. And it turns out that the way that your DNA is read can be changed through environmental factors. Now, this is still a very nascent sort of field. It needs to be studied in a lot greater detail, but it turns out there is some pretty good evidence that things like pesticides or smoking or whatever might lead to different ways that your cells read DNA. And that can lead to changes that are, in fact, inherited by your offspring. And if we are so concerned about the consent of future generations, will we institute similar policies against any of these environmental factors, which seems completely infeasible, but on the off chance that you might be impacting the consent of future generations? And so there's a lot of sort of muddy water here, I would argue, in terms of what actually delineates heritable versus non-heritable genome editing. So what was the response from the scientific community? Their response was what they called a moratorium. They said, this was back in 2015, the most prominent researchers in CRISPR and in other genetics and related fields said, look, we need to put a pause on all germline editing. They sort of still buy into this germline somatic divide sort of as a sort of a black and white issue. And they say, we don't want anyone to edit the germline until there are official democratically and internationally determined guidelines on how germline editing should be handled. It may be that international bodies decide nobody's allowed to do this ever, like cloning, human cloning, which is completely forbidden. Or they may say, well, yes, in certain cases, however, whatever the case may be. But regardless, we need those committees to make those decisions before we can proceed with any sort of germline editing. So despite all these calls for a moratorium, unsurprisingly, one scientist decided to completely ignore, because there's no actual regulation or teeth behind a moratorium. It's just sort of a call, a general plea for everyone to withhold and hold back from doing germline editing. And so There was what's known as the CRISPR baby scandal, which some of you may have heard in 2018, where a researcher in China named He Jiankyu edited 2 twin baby girls using CRISPR. And I won't get too much into the details of the experiment, but basically he did a preventative edit. So if you think about those buckets from earlier, where he edited CCR5, which is a protein that if it's not there, you're less likely, or you're inoculated against getting HIV. That was the idea of this experiment. And this caused a huge furor in the media, among scientists, and so on. So what was the response? Moratoriums. So it's kind of amazing, but this was the response from a lot of scientists. And this is only the scientists, by the way, who thought there should be a moratorium. There were other scientists who never thought there should be a moratorium, or now don't think there should be a moratorium, and that germline editing is perfectly reasonable, and there shouldn't really be barriers to that kind of research. So again, more moratorium, calls for moratorium. This, unsurprisingly, is probably not going to lead to a huge shift in behavior. It didn't the first time. Why would it lead now? It's sort of more of a statement of ethics than it is a regulatory decision. And then just on the subject of He Jiankyu, who edited these babies, not only did he go ahead and do this, not only did he, and there's a lot of backstory to this, not only did he liaise with researchers around the world, some of whom rejected him, some of whom helped him on carrying out this experiment, but there was also a conference held by the most prominent scientific organizations in the world, the National Academy of Sciences from the United States, the Royal Society from the UK, the Academy of Sciences of Hong Kong, etc., who came together and hosted He Jiankyu to talk about his research, which doesn't, to me, I don't know, to me it doesn't feel like a condemnation, but this was sort of the response that was provided, I guess the information he provided was deemed necessary for the public good. But for myself and for my co-author Ayakumi, we wanted to sort of interrogate this a bit further. So I'll get to that in just a second. And unsurprisingly, despite the fact that we had such a rigorous and forceful response to He Jiankyu, there's a Russian scientist named Denis Rebrakov who would like to edit babies just the same as He Jiankyu did, and his plan is to alter the deaf gene, which, as I mentioned earlier, is controversial for many reasons, in egg cells, so in germline cells. So that's going to continue regardless. The idea that they would be put off by a moratorium is perhaps imaginative at best. So there's some, so when I mentioned earlier that there were some of these bodies that were supposed to make recommendations or regulations based on input from democratic in a democratic fashion from the public, from ethicists, from politicians, from scientists, experts, and so on, religious leaders, and so on. There are sort of 2 main bodies that are meant to do this, and they're supposed to be producing recommendations fairly soon, but at the time, in 2018, it was still 2 years away, and so that's why these calls for moratorium sort of arose. And the first group is organized by the World Health Organization. The second group is coordinated, at least in part, through the Royal Society. And these are still international organizations. But when we get into actually who makes up these organizations, we find something kind of disturbing, or at least we found this disturbing, which is that and this is unpublished sort of analysis, just for what it's worth, but the World Health Organization, when we broke down who was actually in that committee, most of the people in that committee are based in institutions in OECD countries. Now, OECD countries are considered developed countries. They're high income. This is sort of their official designation on the world stage. And the reason this is important is that we have to really think about how genome editing not only impacts humanity as a whole and us on an individual level, but also how it plays into global politics. And the idea that a lot of countries in the what we call the global north, which is in many ways the synonym for OECD, although these terms are distinct from each other, have used science and have used politics in the past to take advantage of countries that are not OECD, that are not considered developed countries, through policy, through, as I said, through science, and through the use of business practices, etc., when we understand that is a history that we have to sort of reckon with, and then we look at these committees that are making these potentially species-changing decisions, and that they're mostly made up of people who are people who represent countries that are OECD. We have to think about, is that really the most democratic, the most sort of integrated way of producing recommendations? And by the way, if we look at the Royal Society Committee, the International Commission as it's known, you have the exact same problem. It's even more dramatic. And in fact, in both cases, the United States is really overrepresented in both of these committees, even among the OECD wedge of these pie charts. So I'm just going to wrap up really quickly just to highlight an article that Ayakumi and I wrote last year. It's also known as AJ, so I may refer to him as AJ from here on out. And we put together this article, which was a call for a boycott to reinforce the moratoriums that had already been called for, but to actually give them some teeth. And the ideas, just very briefly to get into what this boycott would entail, are first to withdraw all collaboration, support, and advice from researchers who say that they're going to engage with unethical research. So Heritable, for example, would be one of these people. Denis Rebrakov would be another example, and so on and so forth. To say that if they reach out to you for advice, do not engage, do not help them, and we need to draw a line in the sand on that. The second is that we refrain from supporting conferences or journals that publish any of this information. So far, Heritable's research has not been published, but if his research were to be hosted at a conference, if his research were published in a journal, that we would not support that journal or attend that conference. So that's the idea of that sort of pillar of this boycott. And the third is that we would encourage a peer reporting system. So if you are reached out to by a researcher who is engaging in unethical research, you alert your colleagues and say, look, this person is reaching out to CRISPR researchers, for example, or geneticists, and is trying to get support for this unethical work that they're doing. And so if they do reach out to you, just be aware of that and remember the boycott, remember the rules that we're trying to isolate and prevent these folks from getting support from the broader academic community. So hopefully I haven't gone on too much over my time. That basically sums it up. I just want to thank AJ, as I mentioned. He's my co-author and partner in crime in this endeavor. And of course, the Wellcome Trust, the Gurdon Institute, Churchill College, the university, and of course the Jackson Lab for providing me with studentship so that I can be here in the first place. Thank you.
Wow, that was really, really interesting. Found it very interesting. Well done. There's a round of applause for him. You can just use the rest sign. So thank you so much, Søren. That was really interesting. Yeah, so we'll just go straight to questions. If you have any questions, please feel free to unmute and ask.
I have one if no one else does.
Yeah, go on, please.
So are you aware of any pathways politically to discuss it with the various government bodies in order to put essentially more than a guideline and actual laws to prevent people from doing this if guidelines aren't effective in stopping it?
That's a good question. I would say that, I mean, through the normal, however legislation gets passed in your country, that would be the way that I would recommend engaging with the politics of the situation. But in a way that might be perhaps more efficient, all of the committees that I mentioned, the one from the World Health Organization and the one from the Royal Society, these are meant to be, if you've never heard of them, then perhaps they need to broadcast out a bit more. But again, I think there's a real problem with the interface between science and scientists and the broader community. But those two committees are meant to interface with the public, and so they're often holding open meetings, calls for comments, etc. And so if you are able to participate, go find those committees. Their websites are available online, and contact them. And when they have those open calls for comments and discussion, etc., participate in those, because what those committees recommend will be taken much more seriously, I would assume, than every random citizen who manages to get through to a senator or a congressperson. Now, that's not to say you shouldn't do that as well. You certainly can, but these committees are going to have huge sway over how the international community and governments make decisions on how to manage germline editing in the future. And I should also say, by the way, that I don't want to speak for AJ. He's actually on this call now. I don't want to speak for AJ, but we're not calling for a ban on germline editing. There's some folks who firmly believe there should be a ban on germline editing, which is understandable. That's been regular. People have been calling that since since the 80s. And there's other folks who think that there should be no ban and it should be unrestricted. Maybe ethical guidelines generally, but that it should that research and those therapeutic development processes should continue unabated. So we're not taking a side on either side of that, because there's really good arguments in many of these cases. For example, the Tay-Sachs case I mentioned is, I think, a very compelling argument for therapeutic editing and germline editing. And so we're not taking a stand on that, but we are saying that that if we do want to pause germline editing until these committees can make their arguments, then we need to give that pause some teeth. Otherwise, it's meaningless.
Yeah, great question. Yeah. Great answer. Yeah, go on.
Just one question. So you're saying, so it's more about pausing until there's some worldwide agreement as to what the rules are. But are you happy for still research to be happening on germline editing, or not even research?
It depends. I mean, so germline, again, there's the unbelievable detail that I've skipped over in the course of this presentation, but when you, so for example, prior to these, and actually we wrote about this in our textbook chapter, and as we were writing the chapter, more and more of these experiments had popped up and these papers have popped up, where they were, researchers were on, still violating the moratorium, were editing germline cells, or editing embryos actually, that were inviable. So these were embryos that would never be implanted, would never sort of proceed to the next steps, but there were the recommendations and the rules are sort of even against that. So that kind of editing is, I think, a little bit different than going to the clinic with it. But even then, I think there's a lot we have to really think about in terms of, and there's rules on how many days how many days the embryo blastocyst could grow, and so on and so forth. There's a lot of regulations on that, and people could argue the merits of some of those limitations. But we have to think about what value is this research. So if one of the big criticisms of Hay-John-Hughes experiment is that the technology isn't really ready for use in humans. And so because it's not ready, because the editing was actually not very efficient and didn't work very well, these babies have some CCR5, some cells have CCR5 and some cells don't. It was not, there's a mosaic sort of phenomenon going on there. Because that happened, it really raises questions as to whether or not there's any scientific merit or value in the experiment in the first place. So if there is a good case for doing germline editing, and there are researchers who are doing germline editing in a very sort of measured way in the UK and the US and so on, then there's there's potential value there. But we have to really think about it. Are we in crossing this sort of ethical threshold? Are we at least doing it for a reason, or are we just doing it because we want to be the first? Which is, I think, how a lot of people feel Hay-John-Hughes sort of saw this, which is, I want to be the first to create a CRISPR baby. And now forever in history books, he will be. And I think there's a little bit of that aspect to it as well. But if there's an actual reason to do it, at least in a dish, as opposed to in a clinical setting, I think that's probably something that we could explore if there was a good case to be made for it.
Don't we have a risk of, because germline editing is, potentially very dangerous, that everything is going to be stopped as well on somatic cells editing? Is there a risk that the two will be put together as one issue? And
so it's a good question. I think the potential for, one thing that I know my co-author and I support wholesale is what's known as ex vivo genome editing. So this is where you take cells out of a person, edit them, and put them back. So therefore, we're not introducing edits into the person directly, but we're sort of editing their cells and putting them back, and then hopefully that has a curative effect. And that's actually being tested in clinical trials right now. It's something that's really really interesting and really promising work that I think has a lot of safeguards in place, because you're not editing in the person themselves, and it's not sort of inherited. So I just want to throw that out there as a kind of genome editing that I think is happening now, is happening in the clinic. and I think has a lot of promise, a lot of therapeutic promise. So I'm not so concerned about that. I do think that we need to rethink the somatic germline divide and that definition. I think if we can come up with a better set of definitions, and I'm not sure what those would be, but we're operating on a principle set up in the 1980s. And I think we need to update it for the present day, because as I noted, there's a lot of blurriness as to what it is about germline cells that makes them different from somatic cells. There are some clear differences, but there's also some clear blurring of that line. And then also there are all these other things that we do to ourselves throughout our life, which is completely fine, but we sort of don't have an issue with that, even though it may also impact sort of our offspring. And so if we can come up with better definitions, which I think is something that is an outstanding issue in the field, then I think we'll be able to better regulate this in the future, or at least talk about it than we're able to now. And that should prevent throwing the baby out with the bathwater, if you forgive the expression.
Yeah. Does anyone have a question? Sorry.
I might get in on this. Good to see you, Søren. Thanks for the presentation. So I'm going to put you in a bit of a sticky situation, perhaps, or ask a pretty tough question, but it'd be good to get your take on it. So, given the current crisis that we're in, the credibility of science and scientists has been put to the test with coronavirus. And one of the things that we've seen is that consensus is really hard to achieve, right? And various scientists are sort of pushing various theories and hypotheses and models, and we're seeing it sort of play out in different ways in different countries around the world. So, you know, given what we know as it relates to COVID, are you concerned or are you comforted by the response that we've seen among the scientific community? And how can we extrapolate that into trying to find some form of consensus around CRISPR?
So actually, I might refer you to my colleague. So thanks for that. That's AJ, my co-author on this, on these articles, and in many ways co-author of this presentation. So I think it's a great question. I can tell you that by and large, I feel, and we actually talked about this in our chapter before COVID was a thing, that we're extremely concerned. And the reason for that is, you know, I'm from the States, where, as many people know, climate denialism is rampant, climate change denialism. Vaccine avoidance is massive, and so on and so forth. So there's a lot of, and what, when we're thinking about sort of how did that happen, the way we conceptualized it was that because scientists didn't get out in front of the issue, that vacuum was filled by people who were out for profit, who were able to take advantage of people who were really being hurt by this, by these technologies, or felt they were being hurt by these technologies, or by the science, or whatever the case may be. And so, you know, not to cast aspersions on everybody's anti-vaccination, because there are some people who are generally feel that they've been hurt, even if whether or not the science sort of supports that. And I think that there are sort of opportunists who have taken advantage of that situation. And I think that's the same in many ways with climate change, where there were people who felt like they didn't want to change their way of life. And so what Exxon was telling them about climate change was appealing, or tobacco, or whatever the case is. And so what we argue in the chapter, and what I think hope still believe, is that, and we see this with COVID as well, is that we really need to step out in front of the issue and interface with the community. That's partially why I wanted to do this talk. I could have spoken about DNA repair and biology, and you know, I'm sure some people would have found that interesting, but I think that this is really important if people don't know about it. So I think we need to interface with the public who aren't scientists, with lay folks. And I also, just to put a slightly more positive, and I don't think we've been doing a very good job in the realm of CRISPR, so I'll say that. And genome editing, for a lot of people don't even know what CRISPR is, but it is coming, and it's coming fast. But in the context of COVID-19, I'm actually kind of encouraged to some degree. I mean, a lot of countries have had different responses to the outbreak and so on, but I think it's been encouraging to see how quickly people still turn to public health researchers, turn to, you know, in the States, everyone talks about Anthony Fauci, and of course there's mixed opinions from various political persuasions on him, but I think he was in many ways the face of what was going on and the response. And I think that it's encouraging to see that people were willing to at least accept that, and that science hasn't been completely sort of thrown out. But on the other side of things, I also want to say is that scientists maybe aren't the only people we should be listening to on this. We should be listening to not opportunists trying to write a book or make a buck, but rather ethicists and people who have opinions on this who aren't necessarily scientists, but who have relevant ideas, lawyers, and so on and so forth, who are informed on the issue and have well-argued sort of perspectives on this. Because I think there's not a right answer per se, but that if we are willing to accept expertise, which is more broad than just science, then we can perhaps then we'll actually have a path forward on this. If we don't act, I think the more likely situation is that a lot of folks are going to swoop on this, potentially from the same sort of buckets that we talked about before. Anti-vax folks or whatever will come in and say this is evil and we should avoid it at all costs. So I think that it's a complicated question, but I appreciate it. Thanks, AJ.
All right, thanks. There is a question on the chat box, and it says, in the state of the art, how safe is genome editing? What are the potential side effects, etc.?
That's a great question. So this is actually AJ's favorite topic. There are consequences. So first of all, we kind of think of genetics in such a way that like gene A does thing A, like or protein A does thing A. When in fact, it's probably a lot messier than that. And if we take away thing A, gene A or protein A, then it can't do thing A. So it's sort of this sort of binary thinking. But I think that what we don't really appreciate about genetics is that a lot of different proteins do a lot of different things in the cell. Sometimes they overlap in function, sometimes they're separate in function, sometimes it takes multiple proteins to do a single thing. It's great if we want to edit what they call monogenic diseases that are only the result of a mutation in one gene. But what if it's a result of a mutation in multiple genes? Can we edit all of those genes at once? Can we do so efficiently? And so I think that we don't really know enough, which is another reason why perhaps it's not the time for germline editing. So that's one thing. Another thing that's really important, and the most important sort of safety concern about this is that, or one of the most important safety concerns is off-target editing, and this is the idea of specificity. And so I won't get into the details of it, but basically, if we want to edit gene A, there's a chance you might accidentally also edit somewhere else in your DNA. And that's a real concern. And that has not been addressed in a meaningful way. It's been studied a great deal by fantastic researchers, but it hasn't been ameliorated. There's always a chance that you'll end up editing where you don't need to, which is partially why I like, as I mentioned earlier, ex vivo therapies, because you can edit those cells, check and make sure you didn't edit anything accidentally, then put them back in the body. If you edit in the body, you may edit something somewhere that you don't mean to, and who knows how that sort of might turn out. And then there's a lot of ways of checking for those accidental edits that are quite robust at this point in time, but are still being developed. So there's a lot of sort of those parameters. And then the last thing I'll just say on safety is that we have to think sort of beyond just the biological side of it or the genetic side of it, which is that if we introduce a mutation into a population, if we introduce it into a human population or an agricultural population or livestock, how will that affect future generations of that species, and how will that affect the ecosystem and the biodiversity of that environment? And so these are all things that we have to sort of take into consideration as we consider moving CRISPR from just a sort of a research tool into an actual in vivo tool for making changes in organisms and whole organisms.
Cool. And there is a follow-up question to that. So then what about the accuracy of the intended edit at the moment? How accurate?
Yeah, so that's sort of what I was getting at with this idea of off-target effects or specificity. It's hard to answer that question specifically. It turns out there's a lot of things that can affect this. So for example, if you try and edit a particular place in the genome, depending on how the DNA is folded, the CRISPR might not be able to get in to make those edits. That's one thing that can affect that. And different people have DNA folded in different ways. Different organisms will have very different ways that DNA is folded, also different genomes and different sort of contexts there. So there's a lot of sort of factors in that sense. How CRISPR gets into the body or gets into whatever it's trying to edit can affect this. what kind of protein you use to do CRISPR. There's a lot of them. I just mentioned Cas9, but there's a lot of different kinds of Cas9. There's modified Cas9, things that have been changed in various ways to make them more efficient or more specific. And so it's hard to sort of say generally. Most CRISPR edits will happen if you design them, and you can design and sort of predict how they'll behave, which is an important aspect of this, which I didn't really get into, but you can predict how a CRISPR system will, so if I say I want to target gene A and I want to use this CRISPR system, I can use a program to predict how well it's going to target that location and how unlikely or likely it is to target places I don't want to target. And as these algorithms develop and as these sort of predictions develop, we can sort of eliminate at least some of the risk, but there's always going to be that sort of level of risk involved. And it's like with any medicine, we have to consider whether or not this is a risk we're willing to take if it means curing something that's incurable or that can lead to a fatality of some kind. So I think that it's perhaps not at the stage that we can even make that assessment, but at some point we will have to make that decision. Is the risk to the biodiversity of the population, is the risk to the ecology of the system, is the risk to the health of the person worth using this sort of CRISPR intervention?
Yeah, great. There's some questions here. So you mentioned change. What are some of the complications CRISPR scientists have faced in their research that are related to climate change, if any?
So I think that's interesting. So when I mentioned climate change earlier, what I was getting at was more the idea that the resistance to accepting that climate change is a reality that we all must deal with is something that I fear will is sort of a part of a larger continuum of not believing scientists when they lay out the data and the information. So that's sort of where that came from. I actually do think that there's some interesting ideas in CRISPR to sort of help address climate change. I wouldn't turn to CRISPR as the first port of call at all for this, because I think there's a lot of other things we can do. For what it's worth, there's about 100 companies responsible for 70% of the global emissions that are causing climate change, and I think we'd probably start there if we were wise. But on the subject of, for example, agriculture, if we get to a stage where the deserts have expanded dramatically and there's no water anywhere and we still need to grow crops, there are drought-resistant crops that we can develop using CRISPR, and that's actually already being studied and researched. And so there are ways that we can sort of, it doesn't address climate change per se, but sort of gets around some of the ramifications of climate change if we don't deal with it. So in that aspect, there's some of that going on as well. And then I would say just generally, this is completely personal opinion, but there is a lot of feeling at least in my lab and in other places where as we look at the impending sort of situation with climate change, it makes us feel like whatever we're doing in the lab feel kind of pointless. And I'm sure other people feel this in their jobs too, because we're like, what's the point of curing cancer if the world's on fire? So that's kind of grim, but it's sort of a feeling that some of us have. But in terms of direct action that CRISPR can be used in that context or how it's been affected, I would say that agriculture would probably be one of the number one ways to think about it.
Wow, great. Thank you so much. I don't know if anyone has any more questions.
Yeah, just to go back, sorry, to what you were saying about like time and how people, don't want to change their ways, and that's why. Yeah. Isn't CRISPR a risk also of people going, oh well, I can just edit this in terms of more preventative or cosmetic? I can just edit this. Why would I eat better? Because some like. It is proven that even with some genes that can, be more like dangerous for someone towards cholesterol or stuff like that, if you eat a good diet, then you're very much not likely to have problems with your cholesterol.
Fantastic point. Thank you so much for bringing this up because it allows me to plug something. So in addition to my work in the lab and so on and so forth, I'm also an editor at Science for the People magazine, and this magazine focuses on radical science, which is the sort of this idea that science and politics this idea that science is siloed away from the rest of society is ridiculous, and that we should acknowledge that our work gets used in ways that we may not agree with, and we should engage with that and try and sort of shape that conversation. So I actually have an article, which won't be published for quite some time, but keep an eye out for it. It'll exist at some point, on this exact idea, which is that we can't, one of the ways I'm sort of framing it is that we can't CRISPR our way out of poverty. We can't CRISPR our way out of public health sort of crises. And so you've seen this already. So there's already, there was a company not too long ago, maybe last year or the year before, that was editing, and they still exist. I forget what the name of the company is, but it's in the States, and they were editing food or crops that were then being served in restaurants in the United States. And they did this sort of without people's knowledge, which is kind of crazy to me. And also, I think sort of bypasses regulatory rules in the United States. But anyway, so they sort of served it through a restaurant. And I guess that's maybe how they got away with it. And their argument, it was unbelievable on their I wouldn't say this is widespread, but this did happen, or this you can look it up. I think it was in Ohio or something like that. Their argument on their website for why they were doing this was they had created a version of corn, and it's not even a question of safety, by the way. I'm sure these were safety, but although, you know, who knows? But the idea was that it wasn't safe, but the question of why do we need to use these foods? And their argument was, life expectancies in the United States are dropping, or they're not increasing. And this low fats crop that we're now using, that we've edited with CRISPR, will help prevent or help sort of address that problem, because it'll help fight obesity. This was crazy on many levels. For the first reason, obesity is not the reason that life expectancy has been dropping in the United States. There's a lot of things like opiate addiction and so on. There's also things like poverty and social determinants of health and so on, which are extremely important. And to use this as a legitimate argument for why you should CRISPR food is just wrong. I mean, to me, it's wrong. And it probably worked really well in like an investor meeting, and so they put it on the website. Anyway, it's no longer on the website, but you can still find it via the Wayback Machine. And so I'm going to be talking about this and a few other ideas, which is that, like, you know, if one of the things Bill Gates wants to do, which is actually amazing to me, is he's gotten on the gene drive hype, which is a CRISPR system that you can introduce into organisms. And the way this works is that basically you can wipe out a large population of organisms very quickly. And so they wanted to use this in Africa to get rid of mosquitoes that were carrying malaria. The problem is, A, the consultation with local people was non-existent. B, we don't know what the ecological consequences of this would be. Obviously, mosquitoes serve a purpose. And C, mosquito nets exist. Why are we editing mosquitoes to do? It's just, to me, it didn't make any sense at all. And so there is absolutely a risk of people running away with this. And that's absolutely something I want to talk about, because I think it's really, really important that, you know, just because we can doesn't mean we should. And so that's a real risk. And yeah, so thanks for the question. And I think it's something that I've been thinking about a lot, and I want to write about in some detail moving forward, because I think that there's, you know, just one last example, the two babies that were edited in China. Yeah, you can prevent them from getting HIV in the future through CCR5 edits, but also, like, condoms exist and other, you know, and drugs that can prevent you from passing HIV onto your children if the concern is in utero. So why are you doing this? What's the argument for it? Even less the case with the gene for deafness. If you can edit it, should you? I don't know that makes a whole lot of sense. In the case of Tay-Sachs, yeah, I think it makes a lot of sense, but we have to really think about these things, because I think just because we can, as I said, we shouldn't.
Yeah, and that brings us to a very good end. Just because we can doesn't mean we should.
Exactly.
So much. It is really a very, very nice presentation, and I'm sure you all agree with me that Søren did justice to that talk. So if we could just give him a round of applause to say thank you.
Thank you.
Thank you.
Thank you.
Thanks.
Thank you very much.
Yeah, thank you so much, Søren, for doing this talk.
No worries. Thanks, everyone. And if anybody has any further questions, feel free to email me. My email is right here on this slide.
Great, thank you.
I'll definitely reach out to you, because yeah, I think there's so much to do.
There is.
Yeah.
Thank you so much, everyone.